Welcome and good morning to this formal presentation and webcast of Redwood Pharma's Phase 2 trial of RP101 in patients with moderate to severe dry eye disease. I'm Martin Vidaeus, I'm CEO of Redwood Pharma, and I'm joined this morning, March 12th, 2020, in Stockholm, Sweden, by Professor Gerhard Garhofer of the Medical University of Vienna, Austria, who is the principal investigator in the trial. This morning, Redwood Pharma issued a formal press release announcing the positive top-line results for our shareholders and the broader investment community. This presentation will expand on those top-line results and give more detail into the trial and its conclusions. After the presentation, we will have a question and answer period for about 10 minutes, and we welcome questions for those who are in attendance and for those who are watching on the webcast, who can pose questions digitally through the webcast interface. And with that, we will now start the presentation. I'd like to first give a brief overview about dry eye disease for those who are unfamiliar with the disease. Dry eye has several different causes. It's a multifactorial syndrome that there's many different causes of it, and it can be broken down into two categories, both evaporative and non-evaporative. And that depends really on the glands that are affected in part in the production of different components of the tear film, which can cause different variations of the disease. There are risk factors of dry eye, such as the aging, dry climates, changes in hormone levels, and symptoms and manifestations can be varied. They result in an inadequate tear film, and primarily it is tearing, foreign body sensation are the most prevalent symptoms, but there are a host of other symptoms that also can manifest itself. The market is large for this unmet medical need. There are an estimated 344 million sufferers globally, and it is the most common reason why patients go see an ophthalmologist, dry eye being that reason. We estimate that there are approximately 10 million women in both the US and the EU that suffer from this disease who today have an unmet medical need. Today, there are no adequate therapies really to help these sufferers. The first line of therapy today are artificial tears. Those are standard saline solution products that you can buy at the pharmacy and take them up to 10 times a day if you really have a more difficult case, moderate to severe cases. After the first line of therapy, the traditional products that are used today are to treat inflammation. And those products today, such as Restasis and Xiidra, that deal with inflammation, take a long time to actually treat the resulting inflammation. So there is today a need for new products. And that really is the underpinning of our product, RP101, and why we strongly believe in this product development program. I'm going to tell you a little bit about the product itself before I hand over the presentation to Dr. Garhofer. RP101 is based on two components. The first component is estrogen. It's an active pharmaceutical ingredient that is a well-known estrogen analog. Estrogen is found in over 500 different products globally with a known safety profile as well as an efficacy profile. What we're doing is applying a topical low dose in order to achieve a therapeutic effect. The other component is IntelliGel, our drug delivery system that controls the release of active substances up in the front of the eye. And what we're trying to do in this case is keep the active substance on the eye longer to help improve residence time, perhaps increase bioavailability, perhaps reduce symptoms that are associated with other drugs, say, for instance, that stay on the eye longer. So our idea now is to improve convenience by applying RP101 once or twice a day, whereas other products are administered more times a day, upwards of 4 or more. The medical hypothesis is basically a change in hormonal balances within women after menopause, that they will then see a drying of tissues as well, and have discomfort in the eye. So we're targeting an underlying biological mechanism for the treatment of dry eye disease by restoring the tear film through applying a low dose of estrogen to the front of the eye. Today, there are similar therapies for diseases based on similar physiologies, such as vaginal atrophy, where estrogen today is used as a topical therapy for the treatment of the disease. So we're looking at similar cell types, the drying of membranes there, which has then led to estrogen being applied as a local therapy that is now serving the segment. And what we are doing is basically piggybacking on the same medical situation. So what I'd like to do now is to hand over the presentation to Professor Garhofer, who is our principal investigator in the trial.
Thank you very much, Martin. Good morning to everybody. I'm happy to present the results of the RP101 clinical trial phase 2. Very briefly about the design and the objectives of the trial. It was a multi-center, double-masked, placebo-controlled trial with 8 recruiting centers located in Austria, in Germany, and in Hungary. We had 4 treatment groups, 3 active groups, and 1 vehicle placebo groups, and give you more detail in on the next slides. We also tested once and twice daily instillation, and the active treatment period was 90 days. As usual for a phase 2 trial, the main objectives were assessment of safety and of efficacy, and the further objective, of course, was to find about dosing and appropriate doses for further development. Very briefly to the endpoints. The endpoints of the study were to evaluate the clinical efficacy during and at the end of the treatment period with RP101. The primary efficacy endpoint was reaching 10 millimeters on the Schirmer test type 2. This is the Schirmer test with anesthesia. Secondary endpoints included tear breakup time, which is usually a measure of tear film stability. We used the Sandy questionnaire to get information about subjective parameters, and we measured additional outcome parameters like foreign body sensation, tearing, and other variables that are known to be associated with the clinical picture of dry eye disease. Finally, we assessed safety based on the frequency of adverse events. My first result slide is related to the demography and the patient disposition. You can see here that we have 3 different active groups. One group 1 consisting of 0.05% of RP101 installed in the morning, and 0.05% in the evening. Group 2 consisting of 0.1% in the morning and placebo in the evening. And group 3 of 0.1 in the morning and 0.1 in the evening. Group 4 consisted of administering placebo at the morning and in the evening to obtain double-blind conditions. The last column gives us information of the overall demography. You can see here the total number of included patients was 104. The average age was 62.9. We had dropout rate, overall dropout rate of about 25%, which was, however, mainly derived to group 3, which had a higher dropout rate compared to the other 2 active groups. Well, this is the present the slide presenting the main outcome variables, the Schirmer test. You can see the value of the Schirmer test on the x-axis and on the y-axis the time of treatment. The red line indicates the 10 millimeters cut off for the primary endpoint. And what you can very nicely see is there is a pronounced decrease in all groups in Schirmer values, which reaches level of significance already after 14 days of treatment in 3 of the 4 groups. And it continues to grow until day 90. So in summary, we have a pronounced increase in Schirmer's test, starting at the values from approximately 3 to 5 and almost doubling at the end of the study period. So the conclusions regarding the primary endpoint are as follows. The study almost reaches the primary endpoint goal. RP101 more than doubles fluid production in certain cases from a very low median baseline. We have statistically significance as early as 14 days for all active groups except the placebo group. Schirmer test results very nicely confirm that it is a variable suitable as a primary endpoint for further product development. I think our results show also that once and twice daily administration is an appropriate way to use RP101. And finally, IntelliGel per se has a strong effect and increases Schirmer test also very nicely. Second part of the results, this is breakup time. As I have said before, breakup time is a measure of tear film stability on the ocular surface. And you can see that in the treatment, in all 4 groups, there is a pronounced increase in breakup time after starting the treatment, which reached level of significance 30 days after treatment and continued to grow until day 90. So the conclusion for the secondary objective endpoint is that breakup time shows a significant effect as early as 30 days in all groups. Schirmer and breakup time increase in parallel, which again strengthens the clinical hypothesis and the conclusion there is a positive effect on the disease. Subjective symptoms are crucial in the interpretation of dry eye studies, and so I'm going to switch to subjective parameters now. These are measures for tearing and foreign body sensation, 2 very common sensation in dry eye disease. And you can see at the first glance, this is assessed using a visual analog scale, that there is a decrease in both tearing and foreign body sensation throughout the study period, reaching its minimum at day 90. These are results from the so-called SANDI test. The SANDI test is a questionnaire test, a standardized test for assessing symptoms in dry eye disease. And again, you can see here that there is a pronounced decrease of symptoms, reaching the minimum of symptoms at the end of the treatment period, 90 days after starting the administration of the drug. So the secondary endpoints, the secondary subjective endpoints conclusion are as follows. Foreign body sensation, itching and tearing symptoms all show significant improvement as early as 14 days. The SANDI test showed significant improvement as early as 30 days after treatment start. And after 3 months, all active groups showed an improvement for all symptoms. No statistical significance has been detected between groups. However, it had to be mentioned that the study was not powered to detect differences versus groups. Finally, safety. This is an overview about our safety measures. Safety were assessed by the frequency of adverse events during the study. You can see that most observed adverse events are adverse events that are related also to the disease, that are also symptoms of the disease. The most frequent observed adverse event was blurred vision, followed by eye pain, eye irritation, ocular hyperemia, and swelling of the eyelid. Finally, foreign body sensation. So basically, our safety conclusion are that we didn't observe serious adverse events during the study. The adverse events that occurred during the study were mild and transient. Most of them were, as you saw in the slide before, related to the disease itself. We didn't find any appreciable change in visual acuity or intraocular pressure in neither of the groups during the 90-day of use. And based on this data, we conclude that RP1A1 is regarded as safe based on our phase 2 study results. And with that, I want to leave the summary up to Martin.
Thank you very much, Gerhard. Thank you. And to wrap up our presentation today, I just want to say that we are so happy within the management team and the shareholders. I'm sure in Redwood Pharma that the results turned out as great as they did. I want to thank also Gerhard Garf for really steering the project. We can summarize by saying that objective parameters show statistical improvement, and that I think is a really powerful aspect, as this will be required once the product is going to be seek regulatory approval for market authorization. We will need to confirm that. We will also need to confirm that there are subjective parameters that support the case as well, and here we show improvement where some are statistically significant. And how great is that? We have both objective and subjective parameters that show improvement. And also, a surprise was IntelliGel's positive effect on Schirmer test, which I think potentially expands the market for all sufferers of dry eye, not just women, but also including men at all ages. And that is something that we will have to look at more deeply within the company. And RP101 more than doubles tear fluid production, where fast onset was seen as early as 14 days. I think this is a really compelling story that we can show to patients in the future, that with RP101, that a quick onset can help these patients. So we feel that we are in an excellent position to continue our commercial efforts in finding development partners for the later stage here of clinical development. We're very positive about the future. And with that, I will now then end the presentation, and we'll take a 2-minute pause, and we will accumulate questions that have come in over the internet and within the room, and we'll get back to you shortly. Welcome back. We've now accumulated all the questions that have come in over the net, and we'll give ourselves a few minutes to answer these questions. The first question comes from the audience. How do the results stand against competitors like Restasis, for example? And why don't I ask you, Gerhard, to answer that?
Yeah, I think the main advantage of RP, or the main strengths of RP1A1, is certainly the fast onset of action. We saw a statistical significant increase already after 14 days of treatment, which is a strong sign of efficacy. How it compares to other treatments, to finally answer these questions, head-to-head studies would be necessary, which we don't at this time of development, we don't have. But I think that especially if the fast onset and the strong effect on the Schirmer test is are the two strengths of RP101.
Very good, very good. The next question is, do you consider IntelliGel to be validated? I think I'll answer that question. I mean, IntelliGel has shown now in the trial that it is a very good vehicle. And it had even some benefit in terms of the Schirmer test. There are still more questions that need to be answered, but I think that we have a really positive outcome here. From a commercial standpoint, I think this is even more interesting to potential partners in terms of combining IntelliGel with other active substances to develop new products around. So we are learning more, and we need even more information. But I think with every day, we are seeing that this potential is being turned into reality. Okay, a third question. What is the route from now on? Are there different interests for RP101 and IntelliGel? Well, the company is in active business development mode. We have many discussions going on with global and regional and national pharma companies regarding both RP101 and IntelliGel. I think that these results just strengthen, again, the case for this as a commercial therapy. We look forward to sharing this information with commercial partners, and we will be doing so immediately. Okay, moving on. A question, and a very pertinent question is, how does the coronavirus affect investment interest in general within the pharmaceutical industry and within ophthalmology? And I have to say that yes, this is a complete unknown for us. We can just look at make a comment about the general markets and say that we see general indices fluctuating heavily. I cannot make any pronouncement as to how the investment community will react to our investments in light of these large, more macro problems from a global scale, even from an economic scale. We hope that things will obviously stabilize, and we won't see a large negative effect. And then another question was, what's your perspective on the difference between placebo and the active groups? And can I give that to you, Gerhard?
Yeah, absolutely. I mean, as you pointed out correctly, also the vehicle, the IntelliGel, had a strong effect. We saw that in the phase 2 study. However, in interpreting the results, we need to consider that the study was not powered to detect differences between the active ingredient and the vehicle. And this needs to be figured out finally in a phase 3 study, which is then adequately powered. I think we could identify a suitable outcome parameter for Schirmer test, also for subjective assessment of patient's subjective parameters. And with that, I think the rest of the question has to be solved in phase 3.
Very good, very good. And the last question we have, and I want to thank everybody on the internet who has engaged with us, is considering your successes today, is the company's direction to seek a partnership, or do you have any other plans? I can say from my standpoint as the CEO that these are very good, very positive results that I think just confirm what we have been thinking from the start, hoping that this project would reach. We have a really valuable asset here, and I'm a little bit reluctant about saying which way we're going to go. We have, whether it's going to be, whether we're not going to be open to entertaining other strategic options. The intention at this point is to continue with our efforts to seek a licensing partner to help us go into phase 3 with this product. So with that, that ends our webcast for today. A big thanks to all of my colleagues at Redwood Pharma that have been instrumental in shepherding this project through the end of phase 2. I want to thank Professor Garhofer here, who's been an incredible resource in the work. And with that, we look forward to a positive 2020 and realizing the asset value of RP101 and IntelliGel. Thank you.